While heterotrophic organisms gain energy through respiration, a bulk of the energy that is produced on planet Earth is produced through the process of photosynthesis. Photosynthesis is a process that autotrophs use to gain energy from light coming from the sun. Let's look at that process in more detail. Here's a brief overview of photosynthesis and the products it makes. You can see that sunlight, carbon dioxide, and water are all used to create sugars. Those sugars, or carbohydrates, are using carbon that comes from the air as carbon dioxide. As a byproduct of photosynthesis, oxygen is made. Oxygen is complementary to respiration. Respiration requires oxygen and creates carbon dioxide as a byproduct. Here's a reduced equation that represents photosynthesis. 6 CO2 molecules and 6 water molecules are combined to make 1 molecule of glucose and 6 molecules of elemental oxygen. Since we're building a larger molecule from smaller molecules, this process is catabolic. It requires energy. That energy comes from sunlight. In this image, you can see sunlight lighting up the landscape. Water molecules in the clouds have broken up that sunlight into its component wavelengths. You can see that these component wavelengths appear as different colors. These colors are representative of different wavelengths and different energies. A wavelength is the measurement between two peaks of a wave. We call the light that we can see with our eye visible light. But this light is just a small fraction of all the different kinds of wavelengths that exist. Our eyes have adapted to see the kinds of light that are most important in our evolutionary history. Some organisms can see more infrared, and some organisms can see more ultraviolet. The wavelengths that we can see range anywhere from 400 to 750 nanometers. So, the color red is approximately 750 nanometers from peak to peak. You can see that the redder the color, the lower the energy of wavelength. As the wavelengths become higher energy wavelengths, they appear more blue or purple. It's also helpful to note that ultraviolet is a very high energy wavelength. UV are the kinds of wavelengths that burn your skin when you get a sunburn. Infrared is a lower energy wavelength. Many organisms give off infrared or heat energy as a byproduct of metabolic processes. So, within visible light, we have all these different wavelengths and different energy values. Looking at this spectrum of visible light, which colors do you think would be most valuable in converting light into chemical energy? Plants contain many different pigments, but if we want to see all the different pigments, we have to separate them somehow. In this demonstration, a piece of chromatography paper is placed in acetone. There's also a little dot of leaf extract placed below the line. The acetone will run up the paper. As the acetone runs up past the leaf extract, it pulls along with it the different molecules of pigment. The heavier pigments get stopped by the paper first. Lighter pigments or smaller pigment molecules carry on further up the paper. Given enough time, all of the different pigments will separate into individual bands, and you can see all the different kinds of pigments that made up the original mixture. In this case, on the right, you can see that there's no less than 6 different pigments that made up the leaf extract. Here's an example from a real paper chromatography. There's a lot of different kinds of photosynthetic organisms, and they use a lot of different pigments in order to get energy from light. Those pigments interact with different wavelengths of light differently. So you can imagine plants that grow on the forest floor are exposed to filtered light from treetops above. They would benefit from a slightly different mixture of pigments than the treetops. Trees have access to direct sunlight, so they can use a different combination of pigments. Here's the absorption profiles of several different pigments. Each colored line represents a different kind of pigment. You can see here that we have beta-carotene, chlorophyll A, chlorophyll B, physaerythrin, and physocyanin. Let's look at chlorophyll A and try to understand what this graph shows us. According to this graph, absorption increases as we go up on the y-axis. On the x-axis is the wavelength. Our eye can detect wavelengths from about 400 to about 750 nanometers. So, if we're looking at chlorophyll A, we can see that wavelengths between 4 and 450 are absorbed. From 450 to 600 nanometers, wavelengths are not absorbed by chlorophyll A. Those wavelengths are reflected back. Above 600, we can see that there's a peak at around 660. That means that those wavelengths are absorbed again. This would suggest that the colors that are reflected back at our eye by chlorophyll A are somewhere between green and yellow. Chlorophyll B has a similar absorption spectrum. This means that when we look at organisms that use chlorophyll A and chlorophyll B in order to generate energy, they mostly appear to our eye as green or yellow. Other combinations of pigments would further change the color that is reflected back at our eye. But remember, the color that our eye sees is the color of wavelengths that are not used by the organism. Those are the colors that are reflected back. Here's an example of two different kinds of autotrophic photosynthetic organisms as seen from space. On the land, they're the treetops. Off the ocean's coast, you can see waves of greenish blue. The greenish blue color is produced by bacillariophytes, in this case, diatoms. Diatoms are photosynthetic organisms that occur in the ocean. You can see that the color of green is different than the plants. That's likely because the pigments that diatoms use are slightly different than the pigments that are used by plants. These pigments are optimized for gathering light energy underneath the ocean, where light is slightly filtered by the water. Photosynthesis does not occur in all cells in a plant. In this cutaway of a plant's leaf, you can see that there are several layers. There's an epidermis, a layer of palisade mesophyll, spongy mesophyll, guard cells, and stoma. Most photosynthesis occurs within the palisade mesophyll near the top of the leaf. These cells contain chloroplasts. Like mitochondria, chloroplasts were probably once free-living prokaryotes. These prokaryotes were probably most similar to kinds of cyanobacteria, which are autotrophic prokaryotes. These prokaryotes were taken up by eukaryotic cells through the process of endosymbiosis. Chloroplasts contain stacks of structures called grana. These tall grana structures contain flat surfaces known as thylakoids. Embedded within the membrane of a thylakoid are photosystems. A photosystem is a structure that collects the energy from a photon. There are two main stages of photosynthesis. Oftentimes, we call these stages light reactions and the Calvin cycle. As their name suggests, light reactions require energy from light in order to make products. The Calvin cycle is not dependent on light. It is dependent on products of light reactions, but it can occur at any time. Here are some terms that you'll need to know in order to understand light reactions. Light reactions are the steps of photosynthesis where light energy is converted into chemical energy. The photosystem 2 is a group of pigments and proteins that use light energy to energize electrons stripped from water. Photosystem 1 is a group of pigments and proteins that use light energy to energize electrons received from photosystem 2. The electron transport chain is a series of membrane proteins that shuttle electrons. This is very similar to the electron transport chain in respiration. Like respiration, we have ATP synthase, an enzyme that uses the potential energy in a proton gradient to produce ATP. Let's take a look at the light reactions in photosynthesis. Here you can see a diagram that illustrates the process through which high energy molecules are made from photosynthesis. We discussed an electron transport chain in respiration. The electron transport chain helps maintain a gradient of hydrogen ions across the membrane. On the bottom left, we can see that a water molecule is split into a hydrogen ion and an O2. The donated electrons then enter photosystem 2. There, they're excited by light. In their excited state, they pass through an electron transport chain. It's during that process that hydrogen ions are moved from outside the thylakoid membrane to inside the thylakoid membrane. The electron then enters photosystem 1. Within photosystem 1, pigments collect more light energy and the electron is excited again. That excited electron then allows NADP to reduce to NADPH, a product that's used in the Calvin cycle. Lastly, the hydrogen ion gradient is used to power ATP synthase. As the hydrogen ions flow with the chemical gradient, they're pushed through ATP synthase. The chemical energy is then converted into mechanical energy, and ATP synthase is able to turn ADP into ATP. ATP is also used during the Calvin cycle. The byproducts of the light reactions are ATP and NADPH. The oxygen produced by photosynthesis is complementary to the oxygen required for respiration. Notice how the Calvin cycle requires CO2 in order to manufacture sugars. Within the stroma, in addition to CO2, two other chemicals are present to initiate the Calvin cycle. One is an enzyme abbreviated as rubisco, and then a molecule known as ribulose biphosphate, RUBP. RUBP has 5 atoms of carbon and a phosphate group on each end. Rubisco catalyzes a reaction between CO2 and RUBP, which forms a 6-carbon compound that is immediately converted into a 3-carbon compound. In this diagram, only the 3-carbon compound is shown. This stage is known as carbon fixation because CO2 from the air is fixed into an organic compound, in this case a 3-carbon molecule. ATP and NADPH use their stored energy to convert the 3-carbon compound into another 3-carbon compound called G3P. This type of reaction is called a reduction reaction because it involves the gain of electrons. A reduction is the gain of an electron by an atom or molecule. The molecules of ADP and NAD resulting from the reduction reaction return to the light-dependent reactions to be re-energized. One of the G3P molecules leaves the Calvin cycle to contribute to the formation of the carbohydrate molecule, which is commonly glucose. Because the carbohydrate molecule has 6 carbons, it takes 6 turns of the Calvin cycle to make one carbohydrate molecule, one for each carbon dioxide molecule fixed. The remaining G3P molecules regenerate RUBP, which enables the system to prepare for the carbon fixation step. ATP is also used in the regeneration of RUBP. The Calvin cycle has 3 main stages. In stage 1, the enzyme rubisco incorporates carbon dioxide into an organic molecule. In stage 2, the organic molecule is reduced. In stage 3, RUBP, the molecule that starts the cycle, is regenerated so that the cycle can continue. Photorespiration occurs when the CO2 concentration in the chloroplast drops below around 50 parts per million. At that level, the catalyst rubisco that helps fix carbon begins to fix oxygen instead. This is a highly wasteful process, and it limits the ability for rubisco to operate at its maximal rate. Most C3 plants occur in cool, moist habitats, where they can overcome these limitations. 95% of the plants that we encounter are C3 plants. C4 plants avoid this by having light reactions and carbon reactions occurring in separate cells. There is a cost of ATP. Plants like this occur in hot and dry climates. Corn is an important cereal crop and a good example of a C4 plant. About 1% of plant species are C4 plants. In CAM plants, CO2 is absorbed at night, and light and carbon reactions occur during the day. This limits the amount of carbon available to the plant overall. These plants grow very slowly, and often occur in very hot and very dry climates. About 3 to 4% of plant species are CAM plants, including this example, an Opuntia cactus.